let's start off with cell cycle, and first we'll revise the concept of interphase, which we did yesterday as well. Okay, we talked about mitosis. Okay, mitosis has a certain sort of cell cycle, and so there's this preparatory phase which we call as interphase. Then the main event of mitosis has another four phases, and then at last we have cytokinases. Okay, so your cell cycle basically comprises of interphase, mitosis, and cytokinases. So starting off with interphase, we have three more phases in interphase, which are G1, S phase, and G2, and we talked about all of them. And you'll be also asked sometimes to label, okay, which of the following. For example, they can give you G1 phase and G2 phase, and then ask you what phase is this. So over here you'll have to write S phase. Then right after completing G2 phase, you step into mitosis, which is the actual event. And mitosis further has four more stages, which are prophase, metaphase, anaphase, telophase. And right after telophase, you terminate with the mitosis part and you step into cytokinases. Okay, so cell cycle usually comes for 4 marks, and that you have to write the detailing of every stage. You have to write the detail for G1, S, G2, then prophase, metaphase, anaphase, telophase, and then cytokinases. So we discussed G1, S, and G2 yesterday. Okay, interphase is the preparatory phase, or it is a phase which occurs before the actual event, that is mitosis, in which your cell is subjected to prepare itself for mitosis. And how is a cell supposed to prepare itself? It does so by increasing in size, A, and your cell increases in size during your G1 phase. So over here we can determine the fact that your cell is under non-dividing condition at G1. So if you'll see the cell at G1 phase, you'll find that chromosomes are not visible. Rather, you'll see chromatin. And chromatin are the thin thread-like structures. They are not condensed. They are not thickened up. So you call them chromatin. So under non-dividing conditions, you find chromatin and not chromosomes. Okay. So you need to mention this. K at G1 stage, you'll find chromatin, the thread-like structures, and in this phase, your cell will increase in size. Then moving to the S phase, which we said is semi-conservative DNA replication. You will study the entire process of semi-conservative DNA replication in chapter 6, which is nucleic acids. So for now, just try to remember in S phase, your genetic material is going to double up. With S, you need to remember your DNA content is doubling up itself in a manner that we know all the cells except the germ cell has 46 chromosomes, right? We talked about this as well. So what do I mean by DNA content doubling up? I mean to say if my cell has 46 chromosomes or 46 chromatin at this point of S phase, now it will start to replicate itself. That 46 ka double up will be seen in S phase, which is 92. So at S phase, you'll see 92 chromosomes. And this calculation comes in your paper 1. When we'll do the past papers, you'll be able to see कि किस तरीके से you need to find out the content. They give you different sort of numbers to try to confuse you. But if you stick with this concept, your cells have 46 chromosomes, and during S phase they'll double up, so it will become 92. Then you'll proceed towards G2, which is like the last stage of interphase, where ATP production will increase. And why will it increase? Because after this, your cell will enter into the mitotic cell cycle, where it needs lots of energy. So ATP production will increase during the G2 phase, and your centrioles will start replicating. So from now onwards, you'll study deeply about centrioles and the function of centrioles. Because now the major role of centrioles will be seen, how exactly centrioles play a part in mitotic cell cycle. ठीक है? So up till now, do you have any confusion?
मिस,
जी.
जो ये इंटरफेस है इसके अंदर जो क्रोमोसोम्स होते हैं वो सिंगल फॉर्म में होते हैं, जो हमने देखे थे कि सिंगल क्रोमेटिड होते हैं, या डबल जो एक्स
नहीं,
शेप होती है?
सिंगल होते हैं, जो हमने थ्रेड लाइक देखे थे वैसे होता है.
ठीक है मिस।
So now let's move to the process of mitosis. Acha, this picture is very important for you because the question to identify these stages usually comes in, I think, in P2. हाँ, P2 होता है ना. So this is a very favorite question of your CI examiners. So I'll let you know the identification points once we are done with the chapter, because अभी बताऊंगी तो ऊपर से जाएगा. But as far as we are concerned for interface, we can see कि interface में you can see thin thread like structures. You do not see the X format of DNA, which we talked about yesterday. I told you के वो इस तरीके से जो आपका डीएनए शो हो रहा होता है in the X format, it is known as the condensed form. Rather we call it chromosome. You'll see that in later stages, but in interface it's easy for you to identify. But people, with students, do get confused between interface and cytokinases. So like I said, I'll let you know the identification points once we are done with the entire detailing, because I know कि ये बच्चे यहाँ and यहाँ they get confused और उल्टा पुल्टा लेबल करके आते हैं. तो आसान सी चीज़ है, it's not very difficult, you'll be able to understand. So yeah, so Abhi, for now we've just studied about interface. Now we are moving towards the main mitotic cell cycle, which will start from prophase and end at telophase. Between them, we have, like, mitotic cell cycle has four main stages, and we need to know the details for all of them: prophase, metaphase, anaphase, telophase, and then last is cytokinesis. Okay. So now we are starting with prophase. And this slide is highly important for you because you need to remember all the details that are written here. And this slide is essential for your P1s and for your P2. In P2s, you have to write all of this. While in P1s, they give you different MCQs on this information. So I have extracted all the pointers and have incorporated in this slide. So you do not need to worry कि हम क्या क्या पढ़ें और क्या ना पढ़ें. So I'll let you know कि कौन सी चीजें जो है वो ज़्यादा frequently आती हैं. Starting off with prophase, which is the first stage of mitotic cell cycle, and from now onwards, your mitosis has begun. ठीक है, which starts with prophase. So prophase, you need to remember that this is the longest phase. And this question usually comes in P1. Okay, which phase among mitosis is the longest phase? So if they're talking about mitosis, because I said okay, cell cycle comprises of three stages: interphase, mitosis, and cytokinesis, right? If the question is saying okay, which phase is longest for mitosis, I'm not saying cell cycle, I'm saying mitosis. So for mitosis, prophase is your longest phase. Okay? Do not make this mistake. If they're asking you about cell cycle, you write prophase is the longest phase. No, you'll get a zero and then you'll say कि longest phase लिखा था. Longest phase लिखा है under the heading of mitosis. So mitosis the longest phase is prophase. And what exactly happens in prophase, that your chromatin, your thread like thin thread like structures, they'll begin to condense and they'll form chromosomes. ठीक है. So remember this point कि interphase has chromatin, the thin thread like structures, while prophase, from prophase, your chromatin, your thin thread like structures will condense to form chromosomes. And this condensation is not that process that we talked about in biological molecules. Do not say कि यहाँ पे condensation का process हुआ है. I'm just using this terminology to depict the fact कि your chromatin will thicken up. ठीक है, if you'll write thicken up, then that's also fine. If you want to write condense, that is also perfectly fine, but do not mix it up with the process of condensation, because if you say कि यहाँ condensation हुई है, your examiner will assume that you have added, not you have added, you have removed water molecules. So we are not removing water molecule over here, because condensation is a synthesis process which involves the removal of water, while we are not involving water over here. Okay, so this is a different terminology used in a different manner over here. So chromatin will condense to form chromosome A, the most important detail. Next, each chromosome consists of two sister chromatid. Like I explained this yesterday in the structure of chromosome. Okay, the similar chromatin will form sister chromatid. Okay, so if for example this is your chromosome, then this single thread of this chromosome is your sister chromatid. So these are important terminologies because you have to write these keywords in your script. You can sister chromatid, and we'll talk about daughter cell, daughter nuclei, parent cell. तो ये सब चीजें पता होनी चाहिए कि what exactly sister chromatid is. Sister chromatid is the joining. It join two sister chromatid joined to form this chromosome. ठीक है? So this one is sister chromatid. And how were they held? We already talked about the constricted site, which is centromere. So you need to add this detail as well. Okay, sister chromatid join to form chromosome, and they are held at this region. In this region, this constricted site is known as centromere. Then we'll say कि spindle fibers are formed. So I'll explain what spindle fibers are, but with a different example because अगर इसकी English समझा दी तो सारा ऊपर से चला जाएगा. So I have an example for spindle fibers. I'll let you know. But you need to remember this detail as well, that spindle fibers are formed. And at the end of prophase. So they sometimes give you this question, okay, what happens in early prophase and what happens in late prophase. ठीक है, so do not get confused कि हमने तो early और late prophase नहीं पढ़ा था. Your early prophase, it's nothing, it's just a manner to differentiate, since we claim कि prophase is the longest phase. So we say कि prophase की starting में कुछ events हो रहे होते हैं, prophase जब terminate होने वाला होता है तब एक set of different events हो रहा होता है. But we consider the entire process to be under the heading of prophase. So if they sometimes give you this question, okay, what exactly is happening in late prophase, then you need to add at the end of prophase or towards the end of prophase, nucleolus, which is found within the nucleus, the spherical body, it disappears and nuclear membrane disintegrates. So I should have changed this word over here, disintegrate, rather write disappears, because your CI examiners do not accept this word disintegrate. They reject this word. So this is my mistake. You need to write it somewhere with you need to write it somewhere in your notebook as well. Use this terminology, disappear rather than disintegrate, because I used to write this word a lot, and my teacher used to give me zero on this. So this is essential for you to remember that we do not write the word disintegrate, we write the word disappear, although the meaning is quite similar, but CI जो है वो reject करता है, so let's not take the risk. ठीक है, so write the word disappear. Okay, at the end of prophase or towards the end of prophase, nucleolus, the spherical body found within nucleus, disappears along with the nuclear membrane. Because अभी इनकी regeneration भी होती है, they'll reappear. But we'll study कि किस phase में दोबारा reappear होते हैं. But for prophase, you need to remember, but do not write this word कि in early prophase, nucleolus and nuclear membrane disappears. You need to write this specifically कि towards the end of prophase, यानी जब prophase खत्म होने वाला होता है, at that time your nucleolus and your nuclear membrane will disappear. ठीक है, so ये कहीं inverted commas में लिख लें so that you remember कि यहाँ पे हमने disappear का word use करना है, कोई और word ना use करना. ठीक है, so prophase is the longest phase. Then we said that your chromatin will condense to form chromosomes, and how are they formed? They are formed by the joining of two sister chromatid with the help of centromere. Then we say that spindle fibers are formed, and then we say that at the end of prophase, your nucleolus and nuclear membrane will disappear. So let me see कि अगर यहाँ picture दी हुई है spindle fiber की, वरना I'll explain you with a different example. बहुत light से हैं, but समझ आ जाएगा. So you see these dotted lines over here. ये जो dotted lines हैं, you'll find them when your cell is dividing. So these dotted lines are spindle fibers. ये वाली जो ये दी हुई है ना, these are spindle fibers and they have a very important function. तो अभी मैं example दे रही हूँ, you'll be able to understand. Have you seen those places जहाँ पे जो ऊपर नीचे वाले घर होते हैं, अगर कोई रहता है उसको ज्यादा समझ आएगी कि कभी ऐसा होता है कि हमें कोई चीज नीचे से हम ऊपर मंगवा रहे होते हैं. यूजुअली फ्लैट्स में मैंने ऐसा देखा है कि एक बकेट होती है या एक बास्केट होती है, एंड पीपल अटैच रोप विद इट, और उसको वो नीचे डालते हैं. तो जो भी नीचे बंदा खड़ा होता है, कोई सब्जी वाला होता है, उसमें सब्जी डाल दी जाती है, एंड लोग फिर उस रोप से ऊपर अपनी बास्केट खींच लेते हैं, राइट. ऐसा किसी ने देखा या मैंने कोई अजूबा बात कर दिया? ऐसा होता है? यू
जी।
लिविंग ऑन फर्स्ट फ्लोर। फर्स्ट फ्लोर से आपने एक बास्केट जो है नीचे फेंकी। ठीक है, ये बास्केट थी, ये आपने नीचे फेंक दी। इसके साथ कुछ रोप अटैच की हुई होती है, और वो रोप आपके पास उसका हाथ में होता है, वो रोप का जो भी वो एंड होता है वो उसके आपका हाथ में होता है। और फिर आप उसको जब नीचे फेंक देते हो, जब उस बास्केट को नीचे फेंकते हो तो उसकी रोप उसके साथ नीचे इलोंगेट होती है, राइट। एंड फिर जब आप बास्केट को ऊपर लेकर आते हो तो वो जो आपकी रोप होती है वो कंस्ट्रिक्ट कर जाती है, यानी वापस आपके पास आ जाती है। तो वो जो रोप है ना, कंसीडर दैट एज योर स्पिंडल फाइबर। के उसके साथ जो लास्ट में हमारी बास्केट अटैच है वो हमारे क्रोमोसोम है, ठीक है। उसका समझो ये हमारा क्रोमोसोम बास्केट है। ये जो यहाँ पे क्रोमोसोम बना है ना, ये हमारी बास्केट है, ठीक है। एंड इसके साथ एक हमने रोप अटैच करी है, और वो रोप हमारे सेल साइकिल में, दैट रोप इज नोन एज स्पिंडल फाइबर। मेक सेंस? ये बात समझ आई? एग्जाम्पल समझ आई या कोई और एग्जाम्पल दो? ऑल राइट, परफेक्ट। तो स्पिंडल फाइबर क्या है? स्पिंडल फाइबर्स एक ऐसे फाइबर्स हैं which emerge during the process of mitosis at the stage of prophase। और वो क्यों इमर्ज हो रहे हैं, उनका एक फंक्शन है। वो आगे समझ आएगा कि क्यों उनको सिमिलर फंक्शन है। जो बास्केट वाली एग्जांपल है वही सेम फंक्शन उनका वहां पर भी होगा, ठीक है। तो स्पिंडल फाइबर्स की फॉर्मेशन, उसका स्टेज याद रखना जरूरी है, बिकॉज ये आपके एमसीक्यू में भी पूछा जाता है, दैट एट विच स्टेज स्पिंडल फाइबर्स आर फॉर्म। एंड एक क्वेश्चन ये भी आता है कि ड्यूरिंग विच फेज न्यूक्लियोलस एंड न्यूक्लियर मेम्ब्रेन डिसअपियर्स। तो कभी दे गिव यू द राइट ऑप्शन के प्रोफेज, प्रोफेज मार्क कर देते हैं, कभी उसमें कंफ्यूज करा हुआ था, प्रोफेज लिखा हुआ होता है, अर्ली प्रोफेज लिखा होता है, कभी एंड प्रोफेज लिखा होता है। कभी कभी एमसीक्यू आता है तो उसमें फिर आपने मार्क करना होता है एंड प्रोफेज, या टुवर्ड्स द एंड वाला जो भी ऑप्शन होता है उसका मार्क करते हैं। नाउ योर प्रोफेज फेज इज कंप्लीटेड, ठीक है। यहाँ पर आपके क्रोमोसोम बने हैं, और यहाँ पे क्रोमोसोम बन गए हैं, एंड आपका सेंट्रोमियर वाली बात आ गई, स्पिंडल फाइबर वाली बात आ गई। ये जो सब वर्ड्स यहाँ अंडरलाइन हुए हैं फॉर प्रोफेज, दीज आर वेरी इंपॉर्टेंट कीवर्ड्स। अगर कभी प्रोफेज अलग से आ जाता है इसका क्वेश्चन फॉर 3 मार्क्स, तो ये सारी डिटेल शुड बी अ पार्ट ऑफ योर आंसर स्क्रिप्ट, ठीक है। Now let's move towards the other stage, which is metaphase. Metaphase is very easy to remember and it's very easy to identify as well. So metaphase में क्या होता है? Now that your chromatins they have condensed and they are now in the format of chromosomes, what will happen कि if this is your cell, okay, एंड वी नो कि इक्वेटर एक इमेजिनेटिव लाइन होती है जो कि बीच की लाइन होती है, राइट। इक्वेटर हमारी अपनी, हम अर्थ के लिए भी वी कंसीडर इक्वेटर एज एन इमेजिनेटिव लाइन। इट्स नॉट देयर बट वी कंसीडर इट, इट्स द हाफ ऑफ योर अर्थ। सिमिलरली इक्वेटर सेल पर भी होता है, एंड इट्स नॉट देयर, लाइक देयर इज नो लिटरल लाइन ऑफ इक्वेटर, बट वी कंसीडर इक्वेटर which is the half of the cell। वहां पर all of your chromosomes, all of your thick chromosomes or your condensed chromosomes, they'll align on equator, ठीक है। In this manner all of your thick condensed chromosomes will align on the equator of the cell। So this is just the detail that you need to remember for metaphase, and it's very easy to identify in the pictures as well because सब एक साथ align हुए होते हैं, बहुत आसान होता है, ठीक है। इसमें तो कोई मसला नहीं था कि क्रोमोसोम पिछले वाले फेज में इन प्रोफेज कंडेंस्ड, up after the formation of spindle fiber and after the disappearance of nucleolus and nuclear membrane, all of your chromosomes are now aligned on the equator. Then comes your anaphase, which is like the most rapid stage for mitosis. Again, I'm repeating it again and again. Do not make this mistake. Question को अच्छे से पढ़ो कि what exactly your question is demanding. If it's saying कि mitosis का rapid stage कौन सा है, you will write anaphase. But if they are asking about cell cycle, अभी cell cycle का नहीं बताया, I'll let you know. But जब cell cycle का पूछेंगे तो एक अलग phase होगा। अभी since we are on mitosis, तो अभी mitosis का बता रही हूँ। ये mitosis का अगर longest phase है तो prophase, और mitosis का अगर rapid stage है then we'll say anaphase, ठीक है। So anaphase में just remember the fact it's the rapid stage, and now क्योंकि these chromosomes are now on the equator, now they'll split up. And how will they split up? They'll split up from centromere, and सिंस वी नो कि हमारा जो क्रोमोसोम होता है इट्स इन दी एक्स फॉर्मेट, राइट, एंड दिस इज योर सेंट्रोमियर. सेंट्रोमियर इज द कॉन्स्ट्रिक्टेड साइट, राइट. So यहाँ से from centromere your chromosomes will divide. They'll separate from the centromere. So if I'll say, but paper में do not write this. This is just for you to remember. Okay, now at anaphase your chromosomes will become V shaped because they have divided or they have split it from your centromere. तो यहाँ वाले क्रोमोसोम जो इस तरफ होंगे वो ऐसे जाना शुरू हो जाएंगे और जो नीचे की तरफ होंगे वो ऐसे जाना शुरू हो जाएंगे. सो if I say ke your chromosomes will now appear as V shape, do not write this. Again, I'm saying disclaimer hai. This is just an example for you to remember. कि centromere से जब divide हो गया, तो your chromosome will now appear as V shape. ठीक है? So your centromere divided, and your sister chromatids have separated from the centromere. What were sister chromatids? जब two chromatids they join with this centromere side, we say that these are sister chromatids that have formed your condensed chromosome. Okay? So now I'm saying that sister chromatids have separated from the site of centromere. And now your sister chromatids, they'll be now known as daughter chromosome. जैसे ही they split up from the centromere, they form daughter chromosomes. यानी जो sister पहले थे, अब they'll be known as daughter chromosomes. And there's this important function of spindle fibers which comes, यानी अलग से question comes for spindle fibers for 2 marks, and they ask you to write the function of spindle fibers. The spindle fibers is essential for you to remember for cell cycle as well as के अलग से अगर पूछ लेते हैं then also आपको पता होना चाहिए कि spindle fiber कर क्या रहा था. I've given you the example कि spindle fiber कर क्या रहा था. It's the rope which is attached with the basket, and your basket are the chromosomes. तो यहाँ से your spindle fibers were radiating from this stage of prophase. यहाँ से बनना शुरू हो गए थे. अब क्या होगा that your spindle fibers will pull the daughter chromosomes to opposite poles. तो यहाँ पर जो spindle fibers लगे हुए थे with each of the chromosome, अब spindle fiber उसको क्या करेगा, ऊपर की तरफ pull back करेगा. तो यहाँ पर if I show you the picture, उससे और थोड़ा visible होगा. ये देखो मेटाफेज पे I said that all of your chromosomes they have aligned at the equator, right? And we can see these dotted lines that are emerging from these chromosomes. So I said कि ये क्या है, आपकी rope है, and chromosome क्या है, basket है. अब जब यहाँ पर एनाफेज पर they started to divide from centromere, they appeared as V shape. Now ये देखें यहाँ से ये सब divide हो गए, यानी ये सब V shaped होना शुरू हो गए. तो इनके साथ इनके स्पिंडल फाइबर अटैच थे और स्पिंडल फाइबर इनको पुल बैक कर रहा है टूवर्ड्स दी पोल्स. सो दीज आर द पोल्स. ये पोल्स हैं जहां पर स्पिंडल फाइबर खींच रहा है, किसको खींच रहा है, क्रोमोसोम के वी शेप को, विच आर नाउ दी डॉटर क्रोमोसोम्स. यहाँ तक इज एवरीथिंग क्लियर? समझ आया क्या क्या बोला है फेज पे? सो टेलोफेज इज द लास्ट स्टेज फॉर माइटोसिस. अब इसमें your daughter chromosomes they have reached the opposite poles. ठीक है? And why am I saying ये daughter chromosomes है? Because अभी ये कंडेंस्ड है, थिकन्ड अप है, और अभी ऑपोजिट पोल पे चले गए, तो अब यहाँ से आपका सेल भी स्प्लिट हो जाएगा. तो जब एक नया सेल में क्रोमोसोम शिफ्ट हो जाता है उसको हम डॉटर क्रोमोसोम बोलते हैं. तो डॉटर क्रोमोसोम आपका ऑपोजिट पोल पर रीच हो गया, एंड नाउ your chromosomes will uncoil and become chromatin again, which means के उनकी condensed form, उनकी condensed shape has been lost, and it will now again become thin thread like structure, and we call that as chromatin. ठीक है? Then again spindle fibers will disintegrate. Do not write this word disintegrate. Write spindle fiber disappear. ठीक है? ये word rejected, बहुत बड़ा बड़ा rejected है, मुझे अपनी स्लाइड में भी चेंज करना चाहिए था. Disappear. disappear. तो spindle fibers अब disappear हो जाएंगे because now they are no longer functional. Where your nuclear membrane will reform, nucleoli will reappear in each of the daughter nucleus because यहाँ पर आपका अलग अलग होने शुरू हो जाएंगे. So you can see कि nucleus यहाँ पर अलग हो गया, यहाँ पर nucleus अलग हो गया. तो जैसे ही वो अलग होने लगते हैं या ऑपोजिट पोल्स पे जाना शुरू हो जाते हैं, तो न्यूक्लियर मेम्ब्रेन भी वापस आने लगती है, न्यूक्लियोलस भी वापस आने लगता है, और ये क्रोमोसोम्स लग रहे हैं ना अभी, ये अपने आप को अभी थ्रेड लाइक कर लेंगे. तो जब ये थिन थ्रेड लाइक हो जाते हैं तो उसको हम क्रोमेटिन बोलते हैं। वहां पे आपका स्पिंडल फाइबर भी आपको उधर पिक्चर में नजर नहीं आ रहा होगा बिकॉज वो डिसअपियर हो गया है। और उसके बाद वी से दैट योर डॉटर न्यूक्लियाई are genetically identical, which is the main motive of mitosis, that you produce nuclei, you produce genetic material which is similar, which is identical to your parent cell. And this stage leads to cytokinesis. To first understand what cytokinesis is, क्योंकि आप cytokinesis from like the telophase marks the termination of mitosis. Now the last stage of cell cycle is cytokinesis. Cytokinesis में basically क्या होता है, एक मिनट, cytokinesis में यहाँ पर you can see the cleavage of cytoplasm being formed. यहाँ पर at this point yani right after telophase you can see a cleavage that is being formed at the site of cytoplasm. तो दिस इज नाउ इंडिकेटिंग कि यहाँ से सेल जो है वो आपका अलग होना शुरू हो जाएगा एंड फ्रॉम दिस स्टेज वी से कि यहाँ पे साइटोकाइनेसिस हो गया एंड साइटोकाइनेसिस में बेसिकली क्या होता है, योर साइटोप्लाज्म स्प्लिट्स अप and you can say that daughter cells have been formed. So daughter cells usually you'll find smaller in size because वो नए नए बने होते हैं, it's smaller, but with the passage of time वो fully mature हो जाते हैं, but इनके पास अपना पूरा मटेरियल होता है. यू कैन सी कि ये जो क्रोमोसोम्स थे वो वापस से थिन थ्रेड लाइक हो गए, क्रोमेटिन बन गए. इनके पास ये इसका एक ही न्यूक्लियस ये रहा, एक न्यूक्लियस ये रहा. इनका साइटोप्लाज्म टेलोफेज से क्लीवेज हुआ था. सो तो बेसिकली द पॉइंट वेयर योर साइटोप्लाज्म स्प्लिट्स or the point where your cytoplasm becomes a cleavage, we say that this is cytokinesis. So cytokinesis is basically the splitting mechanism of cytoplasm, from which we say that your daughter cells have been formed, which marks the completion of your cell cycle.